This is serotonin, a monoamine neurotransmitter that's synthesized from tryptophan, an amino acid. Although much of the body's serotonin is synthesized in cells lining the intestines, we will focus on the portion that is found in the raphe nuclei. After serotonin is produced, it is dispersed through the serotonergic pathways to the rest of the central nervous system. Here, we see serotonin forming in cell bodies of serotonergic neurons, which are located in the raphe nucleus. The serotonin created from tryptophan then travels down the axon to the presynaptic axon terminal. Here, the serotonin is taken up by vesicular monoamine transporters, also known as VMAT2 in this case. It is carried and stored in the VMAT2 until a signal releases the serotonin into the synaptic cleft, or the gap between the two cells. It reaches the receptors and becomes the ligand to bind with and activate the receptors. These receptors are embedded into the membrane of the postsynaptic cell and are called serotonin receptors, also known as 5-HT receptors. The many types of 5-HT receptors are divided into 7 families, 5-HT1 through 5-HT7 receptors. Except for the 5-HT3 receptors, which are ligand-gated ion channels, the rest of the serotonin receptors are GPCR receptors, or G-protein coupled receptors, as shown in the example model. Once the ligand and receptor bind, the 5-HT receptors couple to either inhibit or activate adenylyl cyclases, AC, through GI proteins, impacting the catalyzing of ATP to CAMP, or cyclic adenosine monophosphate. This cyclic AMP is released into the postsynaptic cell and is used to regulate many cellular processes. Additionally, binding of serotonin to its receptors triggers other pathways, including the phospholipase C, or PLC pathway. Phospholipase C is a membrane-bound enzyme that is responsible for the generation of second messengers, which increases concentrations of calcium ions, resulting in activated calcium kinases, such as protein kinase C. These kinases phosphorylate other proteins that regulate cellular functions. Finally, we've finished with the normal pathway of the neurotransmitter serotonin. Now let's talk about what happens when MDMA or ecstasy is introduced. When the drug MDMA or methylene dioxymethamphetamine is ingested, it increases the levels of serotonin in the synaptic cleft by doing two things: blocking the reuptake and enhancing the release. First, MDMA blocks the reuptake of serotonin by inhibiting the SCRT or serotonin transporters. Under normal circumstances, these transporters monitor serotonin concentration within the synaptic cleft and return the serotonin back to the presynaptic terminal. When MDMA is introduced, the SCRT are inhibited, causing a buildup of serotonin inside the synaptic cleft, leading to overactivation of the postsynaptic 5-HT receptors. MDMA also releases an excess of serotonin, resulting in mood-elevating effects or short-term euphoria. However, this burst of serotonin will fall to a low after consumption of MDMA, leaving the brain lacking serotonin, which can be attributed to the negative long-term effects many experience after a high of MDMA. The end. Thank you for watching.